everyone, and welcome to today's webinar titled Enzymes for Advancing Point-of-Care Diagnostics. This webinar is part of the 7th annual event in the Laboratory Automation Virtual Event Series. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to thermofisher.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during this presentation. You can find that live chat located at the left of your screen. You can also participate by submitting as many questions as you'd like during this presentation. To do so, simply type them into the Ask a Question box and click Submit. In addition to asking questions, we encourage you to participate in our 4 poll questions at the middle of the speaker presentation. Click on that answer when the poll questions are displayed. Now, if you have any trouble seeing or hearing this presentation, just simply click on that Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credit link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speakers, Dr. Agne Alminaite, Product Manager in Custom Molecular Biology Products Management Group at Thermo Fisher Scientific. And joining her is Agne Ziupkaite, Product Manager at Thermo Fisher Scientific. For a complete biography of our speakers, please visit the Presenter tab from the menu at the left of your screen. Agne, welcome to both of you. You may now begin your presentations.
Thank you, and hello everyone, and welcome to our today's webinar. Today, our topic is enzymes for advancing point-of-care diagnostics. Let's jump to our agenda. Today, we will introduce the general concept of point-of-care diagnostics, also talk about enzymes used in point-of-care assays, and why LIA-ready enzymes are required for point-of-care tests. Let's go to the next slide and talk about what is point-of-care diagnostics. Point-of-care tests is a diagnostic test that allows for diagnosis of diseases outside of a laboratory and usually faster than conventional lab-based testing, with same reliable results. This is enabled by point-of-care diagnostic devices that are small instruments that can acquire specific clinical information. Inside such device, a diagnostic assay is performed at a very small scale. Often, this assay is using lyophilized enzyme mixes to deliver a reliable result. What are advantages of point-of-care diagnostics when compared to traditional diagnostics approach? First, before jumping to comparisons, let me mention that regardless of diagnostic approach, stable reagents and accurate results are essential for both type of diagnostic approaches. In traditional diagnostic approach, we usually find tests performed in a hospital or laboratory environment. Often they take several days and require complicated equipment and trained personnel. Such tests doesn't need glycerol to be removed from their composition. When we talk about point-of-care diagnostics, the tests are simple, portable, and durable. same as the devices that help to carry them out. Less expertise is required to perform such tests. Because of aforementioned test characteristics, the use of lyre-ready reagents is needed. This is needed for storage of these tests and devices at room temperature and also for the capillary-based technology that is used in the devices. Now I would like to introduce my colleague Agne Ziupkaite, and she will tell more about what enzymes are used in the point-of-care assays.
Thank you, Agne. Now that we covered the basics of point-of-care diagnostic, we can talk more about enzymes that are used in point-of-care assays. For your assay to be successful, you need enzymes that have these listed characteristics, like sensitivity, processivity, low DNA contamination, inhibitor tolerance, and thermostability. Thermo Fisher Scientific was one of the first companies that saw the need for glycerol-free enzymes in the market. For point-of-care diagnostics to be successful, it needs to be effective at any point of care. Diagnostic destined for clinic, laboratories, and etc. should be simple, portable, and durable. Historically, the reagents used in most molecular diagnostics met few, if any, of those criteria. Lyre-ready enzymes are not only suitable for lyophilization or air drying assays, but also for point-of-care diagnostic when the applications are done with cartridge-based technologies. Now we will cover the short introduction to methods that can be developed while using lyre-ready enzymes. Lyophilization is the process of removing liquids from a material using sublimation, often a long-term preservation. One of the oldest known examples of lyophilization comes from the Andes Mountains in South America. For more than 3,000 years, members of Aymara and Quechua communities have been freeze-drying potatoes on frozen, sun-exposed hilltops. The combination of dry mountain air, strong sunlight, and freezing night temperatures causes the potatoes' liquids to escape as a gas, leaving behind a latticework of carbohydrates and minerals. In the same way, freeze-drying enables Andean farmers to preserve chung. Lyophilization enables researchers to preserve important laboratory reagents. Even though lyophilization is a really old method, it is still used in everyday life. Because lyre-ready enzymes do not contain glycerol, they are easier adjusted in lyophilization applications. The same needed characteristic of enzymes should appear when it comes to air drying method development. Air drying for molecular diagnostic reagents is relatively new method that has just started to take place in the market. This method is based on water removing from the reagents by heating in the ventilated environment. This method is significantly cheaper than lyophilization, but it can be challenging to apply when it comes to oxidation-sensitive enzymes. Lyre-ready enzymes are non-viscous. This attribute is one of the most important advantages when it comes to cartridge-based technologies. Point-of-care device technology can be divided into two parts. The first one is lateral flow-based technology that is done usually on paper. The other one is microfluidics-based technology done in capillaries. For point-of-care diagnostic devices, the reagents should flow smoothly, not causing clogging in the canals. Our lyre-ready enzymes are perfectly suitable for it. Most of point-of-care assays are based on nucleic acid amplification. As we covered in previous slides, how point-of-care technologies can be divided depending on the assay, now we go deeper into the methods that can be developed using our lyre-ready enzymes. Two widely used methods are PCR and isothermal amplification. Before we go deeper on those two methods, it's time for polling questions.
Thank you for your participation. To record your answers, click on the option on the screen. Our first question, do you plan to commercialize a product within the next 1 to 3 years? Yes or no? I'll give you a few seconds to answer. Appreciate that. Now, our next question, if you have developed or are developing a point of care device, is it a microfluidic POC device or a lateral flow POC device? And your choices are microfluidic, lateral flow. You can also choose I don't know, number 3, and then you can also choose both. Thank you very much for participating.
Let's begin with one-step RT-qPCR method. During COVID-19, this method became gold standard in diagnostic field. This method's main and biggest advantage is high sensitivity. Reactions are done in one vial, relatively fast method, and suitable for high throughput amplification and screening. We can offer the products for the whole workflow in live-ready format. In our one-step RT-qPCR portfolio, we have specially created reverse transcription enzyme, superscript reverse transcriptase one-step RT-qPCR. Since we want our customer to be able to develop this method easier, we have prepared optimized protocol for one-step RT-qPCR method that contains standalone enzymes, superscript reverse transcriptase one-step RT-qPCR, and platinum 2 tag hot start polymerase.
I will continue now on different amplification techniques for POC diagnostics. These techniques are isothermal amplification techniques. What does it mean, isothermal? Isothermal amplification is also a nucleic acid amplification, but it's carried out by a different type of polymerases, a strand displacing polymerases that work at a constant temperature. This eliminates the need for thermocycler equipment. This also makes this group of methods different from PCR amplification techniques. The isothermal amplification usually works at temperature range of 37 to 65 degrees Celsius. There is a wide range of polymerases that carries out isothermal amplification, and in turn a wide range of techniques that you can see here on the right. In the next slides, we will talk about LAMP especially. LAMP or loop-mediated isothermal amplification is the technique which became especially popular for surveillance of SARS-CoV-2. Reverse transcription loop-mediated isothermal amplification, a version of LAMP that is used to detect RNA-based pathogens, has become especially popular technique for point-of-care SARS-CoV-2 testing. This technique is uniquely positioned with nearly the sensitivity of qPCR and nearly the speed of antigen-based tests, which is shown here in the slide, positioning the qPCR tests on the left and antigen test on the right. LAMP stands somewhere near the qPCR tests with the number of the copies of genome that it can detect. Thermo Fisher Scientific offers a superscript 4 RT-LAMP master mix for SARS-CoV-2 detection. LAMP-based isothermal amplification is fast and sensitive for detecting synthetic RNA. The Results are shown in real-time readout after 10 minutes. 4 competitors have been used in this experiment. One of the competitors is Thermo Fisher Scientific product containing evolved BST DNA polymerase, shown here in dark red. The reverse transcriptase, which starts the reaction, is superscript 4 in this master mix. And as you can see, the positive detection result comes already at 5 minutes from the start of the experiment, which shows a very fast and sensitive detection of the pathogen. Now we have a time for our second poll question.
Our next polling question: if you have developed or are developing a point of care device, what is it used for? Number 1, infectious diseases. 2, respiratory diseases. 3, sexually transmitted diseases. 4, cancer or oncology. And finally, your last option, prenatal or neonatal testing. Thank you.
Thank you, and let's continue. Here is a list of enzymes for isothermal diagnostic assays that Thermo Fisher Scientific offers. We have a wide range of DNA polymerases, reverse transcriptases, and related products for your isothermal amplification assay development. Let's move to the next section of our talk, and I'm giving the word now for Agne Ziupkaite.
Thank you. So, how can we help you? Firstly, you need to purchase live-ready enzymes that are compatible with lyophilization process. Secondly, the customer should do their own experiments on pilot scale, evaluate if the system is working on customer's facility. The last step is to prepare production scale lyophilization assay. And now is the polling question time.
Our final polling question: would you like to be contacted by a business development manager to help you with your bulk, custom, or commercial supply needs? Yes or no? And I'll give you a few seconds, and thank you so much for participating in those polling questions.
Thank you for your activity on polling questions. And now let's get back to lyophilization process. Since we covered the workflow to develop lyophilization method, it is good to know how this process can be done. As you can see, there are 2 ways: build your own facility or outsource a third party. It is customer decision to choose which path to go with. Thermo Fisher Scientific has a wide selection of live-ready enzymes for various assays that customers want to develop. If you need any of these products or you need a support while choosing the best ones, do not hesitate to contact us via Thermo Fisher OM page. We support customers at different development phases. We can provide products that are attractive, introductory pricing. There are no need for large volume purchases, and we provide multi-year price control. Our MDX assay developer program have several parts in the program. So 2 divided parts are pricing with discount accelerated, mostly focused on regions, and seed program that focuses on instruments. Side letter agreement is advantage for you since it's advanced notification available for change to form, fit, or function, 2-year contract length with 1-year extension option after initial term, and price protection established. Thank you for your attention. I hope this webinar was interesting and informative for all of you who listened. And once more, thank you for your time.
Thanks so much to both of our presenters. I'm sure that all our listeners will agree that they raised some excellent points during both their talks. We're now going to begin the Q&A session. We'll answer as many questions as possible during the time we have available. We've had several questions submitted already, so we'll move straight onto these. Our first question here is for Agne Ziupkaite. What are the differences or benefits of air-dried versus lyo-ready enzymes for POC assays?
That's a really great question. So air drying is a faster and simpler process that does not require lyophilizator equipment and results in products of gel consistency with similar stability and usability parameters as lyophilized products.
Great, thank you for explaining. I can see we've had a second question come in. This one's also for you, Agne Ziupkaite. I'm trying to develop a test for pathogens with an RNA genome. Which reverse transcriptase is best for this approach?
Another excellent question. The best reverse transcriptase depends on the assay that you are developing. If you are working on a one-step RT-qPCR application, we would suggest to use lyo-ready superscript reverse transcriptase one-step RT-qPCR. If the developing assay is NGS, we would suggest to use lyo-ready superscript 4 reverse transcriptase.
Thanks for explaining, Agne. I can see we've got another question here, and for balance, I'll give this one over to Agne Alminaite. Dr. Alminaite, this question says, I'm developing a POC diagnostic assay based on isothermal amplification. What products would you recommend?
Thank you for the question. As you may have seen in the presentation, our RT-LAMP master mix has proven to be sensitive and fast to amplify from even small amounts of genetic material. We used SARS-CoV-2 material, and both superscript 4 reverse transcriptase that my colleague just talked about and Bsm DNA Polymerase components of the mix are available separately and in lyo-ready format. So as well as thermostable RNase inhibitor, which is needed when you work with RNA genomes. And this product can withstand the temperature of 60 and above degrees that is needed for LAMP amplification. So these are the products we would recommend.
Thank you, Agne. I think we will just have time for one final question, but just as a reminder to our audience, any questions that we didn't have time to get to today will be answered offline as soon as possible. Agne Alminaite, this question is for you. Why do glycerol-free products have to be used in cartridge-based POC devices?
Thank you for the question. Cartridge-based microfluidics technology requires reaction mixes and reagents that do not clog the small channels where the sample preparation or nucleic acid amplification takes place. Also that reagents that do not inhibit the readout process in the device. So that's why glycerol-free products are needed in microfluidic devices.
Great, thank you for the explanation. That's all the time we have for questions today. I'll just give a final big thanks to both our presenters for their presentation and their excellent responses to your questions.
Agne and Agne, I want to thank you for your time today and for your important research. Before we go, I'd also like to thank our audience for joining us today. And again, thank you to our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed for 2 years until March 22nd, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.